So now the panel is Medicine and Society, and I have a lot of speakers here are about to discuss several interesting topics. The first speaker is Sai Reddy, and he's from ETH Zurich, and I believe we will be listening to a very interesting insight about the technologies used in your institute.
Yep.
Please.
Okay, thank you. And thanks for the organizers, and I'm happy to have this very interesting discussion with all of you today. My talk today, I think, will span a few different stretches to it, and I apologize, it might leave you a little bit pessimistic, but it will be but I think it might spark some good provocative discussions. Okay. But before any pessimism, we should actually be pretty optimistic. So some of you might have heard of something called Moore's Law. It's invented or just something developed by a computer scientist named Gordon Moore. And he made an observation way back in, I believe it was in the 1970s, that the power of a transistor count doubles approximately every 18 months to 2 years. And this is a trend that has held almost for 50 years now. And this is essentially kind of covers the idea of how we started with, these computers that were the size of buildings, big rooms that are now computers in our pockets that are, 100 million times faster and more effective. And so really, when you think about where we've come in computing, and we know computing spans so much, that you could arguably say that Moore's Law and its development is one of the greatest examples of human technological innovation, that really very little that we've seen has progressed at this level. And it's persisted, and it's really the backbone of much of how our society function is computing. Now, if we start to look at life sciences, one of the interesting trends that's emerged is that the cost of DNA sequencing. So DNA sequencing is a core tool of engineering life sciences and studying it. And we have seen talks today, talks this week, but also all the time, where DNA sequencing, especially what we call next generation sequencing or high throughput sequencing, are really important. And it's actually since, I believe this is, sorry, I can't see that well, but I think it's like the last 10 years, more like the cost of DNA sequencing has declined so much that it is outpacing Moore's Law. So one of the greatest technological achievements in terms of innovation is Moore's Law, and we're moving faster than that for DNA sequencing. This is essentially making DNA sequencing ubiquitous and very powerful for using in life science research. Now, similarly, not at the same pace. So DNA sequencing here now is in blue, and I believe if I can see here, this is in red, the price of gene synthesis, which is in red here, and then the price of oligosynthesis, which is also DNA synthesis. It's not moving at the same pace of speed, but you do see that the trends are positive, and it is getting faster. So I think it's an important thing to say that the future of life science research has amazing tools that are becoming democratized and accessible and cost effective and everything else. And one should believe that DNA sequencing costs, because of the synthetic biology economic market growing, there will be greater interest in making this faster and cheaper. Okay. So now I want to transition. So that's just why did I talk about that? You'll know a little bit more about at the end. But what were you doing in the summer of 2021? So I'll share what I was doing. Like most people, we're all aware, there was a pandemic that was still sort of going on, depending on what your viewpoint was. Most of us in Switzerland had been able to be vaccinated with mRNA vaccines that summer. I think I received my doses in June and July, and a lot of restrictions were being removed, and life was seemingly coming back to normal. And most people thought pandemic was probably the worst of it was behind us, which is sort of true and maybe not true at the same time. And so I came out and gave an interview to a Swiss newspaper in German, which maybe was not the best idea, considering I don't know German, to give an interview. And just so, since most of you don't read German, you can see that the Daily Mirror picked up what I said. And basically, it created a little bit of online, let's say, energy that I was warning of something called COVID-22. and this was going to be, a big deal. And I got, of course, the inevitable backlash of this being fear-mongering and saying all this stuff. And this is, you can read the date, that's August 2021. Okay, so what am I doing? Why am I talking about these things? Let me show you a little bit more of how much, a little bit of attention it got. I'm not sure the audio is going to work. Oh yeah, here we go.
Wow.
瑞士苏黎世联邦理工学院免疫学教授塞雷迪，他表示，未来有一种叫 COVID-22 的这样的病毒，它结合了印度德尔塔变种病毒、南非贝塔变种病毒和巴西伽马变种病毒，多种病毒产生出超级变种新冠病毒株。
And then a map of infections everywhere, right? Okay, so all right. So I basically have to put a disclaimer here that I'm not a public health expert, not an expert on virology. I wasn't part of this coronavirus task force. I didn't even know German. So what the hell was I doing, giving an interview like this, and putting my reputation on the line? At least that's what my wife said to me when I came home that night, and thought I would need a new job. But actually, there's a little bit more to this, and in a few minutes, I'll exactly show you what I said in the interview, and maybe you'll understand why I felt so outspoken to say it at that time. Okay, so let's come back a little bit. I don't want to give you all PTSD about SARS-CoV-2, but I think we all remember enough about it to know there's this spike protein that's part of the virus. This was a big, this is what actually the vaccines, the mRNA vaccines encoded this spike protein, which is in gray. There's a part of the spike protein called the receptor binding domain. And the receptor binding domain, as the name indicates, is the unit of SARS-CoV-2 that interacts directly with host receptors, a host receptor called ACE2. And that is how basically the virus mediates entry into host cells. And before there was effective vaccines, there was also a lot of development of monoclonal antibody therapy. So basically, these are antibodies here in kind of the light gray that try to target the receptor binding domain and neutralize it because it interacts with the site that engages ACE2. And these are also the types of antibodies we generate when we're vaccinated or when we're or we have natural infection. And so this became a pretty powerful tool used at this pre-vaccine time of the pandemic. However, even though there were a few drugs that were clinically approved, very shortly thereafter, in December, or well, I guess it was late November, almost 3 years ago, I guess, of 2021, we saw the emergence of Omicron. And as studies started to indicate, Omicron had so many mutations that it escaped the majority of neutralizing antibodies. And this is exactly, I think maybe you remember, there was this huge wave of infections with Omicron. We were quite fortunate that there wasn't the same, let's say, severe symptoms. So the hospitalizations were under control in most of, Europe and the United States, but there were some, spots where it was hit hard. In the United States, there were a lot of these antibody drugs were clinically approved, and they were used as supplemental therapy for many people, but they were no longer effective. So they were basically out of luck. And so this made an impact on the treatment at the time. And also there were biotech companies that had invested a lot of resources in developing more antibody therapies, but their therapies were not effective against Omicron, and this was actually a massive loss of revenue and R&D development. So it really caught us off guard. And, the real thing that made Omicron so difficult to handle is that it possessed a massive number of mutations. In fact, it's 50 mutations in its whole genome, but its receptor binding domain alone had, basically 15 times the rate of mutations. And in this, there's even an area that engages directly with the ACE2, which is called the RBM, and this had 11 mutations. It's 50 times the rate, 55 times the rate. So what's going on here? And it's now, I think, maybe intuitive, but basically these sites drive mutations because they drive escape to antibodies because they engage directly. This is the site that actually engages neutralizing antibodies. And as a virus can create immune escape, it allows it to basically proliferate and propagate. and while maintaining its ability to infect and target ACE2. So this immune selection pressure is one of the driving forces of viral evolution. It's pretty clear that this is empirically there, and many people have understood this in other types of viral diseases. Okay, so one of the questions that we had, is it possible to predict these mutations? Is it possible to know, if you know what the selective pressure is, then maybe you can identify what mutations are driving escape. And so in my academic research group at the time, we started to develop a technology where we tried to address this. And this is a very technical, so I'm going to go through it very briefly and kind of on a high level. But the idea is that you take the receptor binding domain, the protein. So this is not the virus. So there's no living virus. It's just the protein that interacts with neutralizing antibodies or this ACE2 receptor. And we create a lot of mutations. We figured out ways to target it. And then in the laboratory, we create synthetic mutations. And then we screen these mutations. And usually we'll screen in the range of 100 million. Okay? But the potential number of mutations that can exist is much larger. In fact, one would argue, if you do the calculations, it's an astronomical number, something like 10 to the 40, because of combinatorial protein diversity. And so we do these experimental measurements using a technique called yeast display and flow cytometry, which allows us to identify mutations that escape binding to ACE2 or maintain binding to ACE2. And we're mostly interested in mutations that maintain binding to this host receptor, because then those could be relevant, because those could, in theory, be a functional virus. And then the mutations that bind, we then screen on whether they escape these neutralizing antibodies. And at the time, there were some of these where we had several clinically approved neutralizing antibodies, and also other ones that were reported in the literature, and they covered different classes. There's different regions of SARS-CoV-2 of this receptor binding name. We call them class 1, 2, and 3. And then we screen for mutations to escape that. And remember, we're only screening about 100 million mutations in total, or variants, but the total sequence space is on an order that I don't even know what to call it, like in the 10 to the 20s. So we actually then have to use all of this, and we then use deep sequencing. So here's where it's super key. We use a lot of deep sequencing of all of these populations, and those deep sequencing data is effectively training data to build machine learning models that, with those machine learning models, you then can predict if any sequence variant appears, whether it would bind to this antibody or escape it. Okay, so it gives you, and also if it binds to ACE2, so could it be a functional viral variant? Okay, so that's it. So we have a model that can do this, and we did a lot of validation to show that the model was quite accurate. And then the most kind of, let's say, fun data to show in some ways was that basically we did in silico evolution. So we took the beta variant at the time. This is a variant that had 3 mutations, originally discovered in South Africa, but had spread globally. And we said, what if we put multiple mutations on a computer, and we ask our machine learning model, does it escape or bind? And red means bind and blue means escape. And these are 4 different therapeutic antibodies that were clinically approved at one point. And you can see that a couple of these antibodies are very robust on top here. So we had 1 mutation or 2 mutations. And so just adding 2 mutations to beta, you actually start to see some blue here across all 4 antibodies. What that indicates is there are mutations that can escape all of the kind of types of antibodies that were known at the time. And then if you zoom in on these are synthetic variants that escape these class 1, 2, and 3 antibodies, and they had mutations in these positions. Now in blue, these were known, 417, 484, and 501. Those are present in the beta variant and also the gamma variant as well. But 493, 409, 8, and 505, those are positions of this receptor binding domain in which there was never any mutation reported in the literature. And you may remember there were researchers investing quite a bit of resources to sequence SARS-CoV-2 genomes. There's probably 100,000 genomes sequenced at the time of SARS-CoV-2, but these mutations had not been identified anywhere naturally. And we identified this in August of 2021. And so this is the main reason why I was quite concerned. And it turns out that when Omicron appeared, all 6 of these positions were mutated. Now, there were more mutations than just that, so maybe we undershot this. But the point is that we could identify the mutations that would drive massive immune escape, immune escape from all types of antibodies. And that was certainly 3 months before the pandemic, the Omicron emergence. Okay, so we've now I want to actually show you why, what I said. And I'll just read through this, the English version from the Mirror. As I said that there would be vaccination breakthroughs in the fall, and cases will increase again, and countries could see tough restrictive measures. I think that was particularly relevant. And then I said it was very likely that a new variant will emerge, and we can no longer rely solely on vaccination. Full context of what that meant is that it would mean we would need boosters. So I said that is why we have to prepare for several vaccinations over the next few years, which are constantly being adapted to new variants. I do want to point out that in this time, the CDC and Anthony Fauci of the US were not recommending boosters yet, okay? And then I said that the beta and gamma variants can partially avoid antibodies, while Delta is much more contagious, but it hasn't yet developed any escape mutations. And I said there'd be some combination is inevitable where beta or gamma become more infectious or Delta develops further, which maybe in parentheses is Omicron, but nevertheless. So really what I said, I don't think it was very controversial in retrospect. Maybe at the time everyone wanted to believe the pandemic was over, but it was really ultimately pretty spot on. This is how I felt before, and this is how I felt after I gave this interview. So, it's not lost on me that I like to compare myself to Leonardo DiCaprio there. Okay, we did a couple other technical manuscripts where we followed up, where we've now done more of this profiling to now Omicron lineages and added. We did kind of a very interesting synthetic coevolution study. Not so important, the technical details. It just shows that we're continuing to explore this. But I want to come to something where I think maybe the future could be, is that it's pretty valuable to do this type of profiling and really start to understand mutations, whether it's existing viruses, emerging ones, and new ones, because it allows us to really design against potentially future variants. It allows more equity and assessment of real time. But there's something else, and I want to point this out, that this is a study that was released in June 2020 from a very great scientist in Bern, Switzerland, named Volker Thiel. And I want to point out something here and highlight it. Using this platform, we were able to engineer and generate chemically synthesized clones of the virus of SARS-CoV-2, which caused the recent pandemic, in only 1 week after receipt of the synthetic DNA fragments. So if you kind of look here, they synthesized the genome of SARS-CoV-2, assembled it, and then put it into cells, and so it shows that it can affect. Pretty high-profile paper. Somehow I think it's sort of been erased from people's memory. And there's no criticism here to the researchers. What they did was really a big effort of synthetic biology. Pretty amazing that they could do this. Now I come back to this, right? So we have this innovation. We know sequencing, DNA synthesis. We also have all these AI tools, computational tools. We also have the fact that there are ways to now synthesize DNA on the desktop. This is becoming democratized. So in a world where technological innovation drives us forward, I want to leave you with this, which is where I think might be a bit provocative, is that we're going to reach a point where it's very possible to design and engineer and synthesize viruses. And these include viruses that can escape the immune system. I didn't design a neural network that could predict escape from the immune system because I wanted someone to use it nefariously, nor did Volker Thiel or his group design something where you can just engineer a virus for any negative consequences. But so researchers working on really good things that are trying to help can create tools that can eventually be ways to create synthetic pandemics. So I think that this is something where, again, this is probably a future we're heading to, whether we like it or not, and do we want to respond to it? How do we respond to it? There's probably good discussions here, so thanks.
Thank you so much. Please. Well, first question, do you have your job still? So that was very, very, very important. Actually, I think this was a very important perspective because every single scientist have been living this kind of stories, especially when speaking with the journalists, because I learned very early that one of the most important things to do is not just talking and leaving the journalists to publish everything, but just reading and reading it back again and editing or whatever, because actually this happens all the times. So one of the perspectives that this works demonstrates is not only the impact that scientific information may have on populations. One of the interesting and intriguing perspectives, and that we are rapidly progressing from a point where we were just counting on cases of diseases. You remember, when we were in old times, we just had reports, surveillance reports, number of cases, and we tried to make predictions. But now we have much more than this. And we have not only the possibility to predict trends, but also to integrate this kind of information in the public health strategies to be implemented in the future. Very powerful. Questions from the audience? 1 and 2, yeah.
I'm going to be bad at the ethics conference and ask a biological question.
Yeah.
How do your very specific SARS-CoV-2 binding predictors compare with using like AlphaFold 3, these much more general protein-protein interaction prediction systems?
Well, AlphaFold 3 is not particularly good at predicting things with antibodies because antibodies have flexible CDR loops, and those really can't be modeled with static structural predictions. So for now, I don't know if if the solution based on structure-based protein-protein interactions are there yet.
Thank you for your presentation and all, and the work that you did. That was really, wonderful stuff. Now, the dual use discussion's been going on for decades, right? And one of the issues that would come up constantly was, okay, what if somebody does come along and synthesize something that's much, much more infectious and much, much more lethal at the same time? And one of the senses of at least some security was the fact that, well, then they will probably kill themselves because whatever they make on their desktop, right, is not going to be in the kind of lab you need to protect yourself from your own product. So there's always that particular dynamic. Do you think that we've moved beyond that sort of limited sense of security of that, if they ever do come up with something, they're just going to end up killing themselves and it won't go any further than there?
Yeah, there's probably two parts to this. So one, it may be part of the goal and willingness to sacrifice themselves. I'm from the United States, and I think we're well aware that mental health combined with weapons can cause a lot of damage. That damage is pretty localized, right? That damage is devastating for the people in the community, but it's localized. What happens when the damage is not localized? I'm going to infect myself and also spread it into some, area, and then we know what happens with viral respiratory diseases and things like that. So that part I don't know. Like basically that could be part of the goal. The second thing is that about laboratories. So I do think like right now it's not a big concern. It requires a lot of specialized laboratory skills. In fact, anybody who can do that would be an awesome grad student. And but I don't think I could. There's probably not people who can do that on their own right now. But the tools are going to continue to develop. And the question is, maybe there's a way to stop state actors. There could be a lot of things like that. But will it reach a point where splinter groups or small numbers of people could do this? And then it may be very difficult to stop it. So maybe I think part of where, if I was ever wanting to take the discussion, is not just prevention, but the response. What do you do afterwards? We saw with the COVID-19, which is, I guess, in retrospect, a little bit of a mild disease. But if it were more lethal, how does the world respond to this? And I think, it's a controversial thing, but I gave a lecture to my course in June of 2020, where I didn't have a statement of one thing that I believed, but I just wanted to point out, using scientific literature, what were some reasons why the hypothesis of being a lab leak were worth taking seriously, just viewing as a scientist, nothing else, right? And I think there was actually a lot of compelling parts to that scientific argument. That was June of 2020. And, last year, the United States FBI put out a report that says, I think the most likely origin was lab leak. Now, there's a lot of stuff going on here, but the point is, we are able to potentially to engineer viruses already. So what happens when it gets easier, when it gets accessible to more people? Yeah.
Thank you. Okay, for the sake of time, let's move on. Thank you so much, Professor Reddy. Very good. Second speaker is Simona Bellegambi. Do you have your slides here, right?
Yes.
Okay. Let me get 1 and 2.
Now, while my slide deck will be set, I want to thank you all, really. I want to express once more how thankful I am to be here with you, to be invited at civil society, because as you will see, I'm the representative of the European Organization for Rare Diseases. And if you were here yesterday, you already heard me saying how important we deem, but I think it's a common statement actually, how important it is that we open the debate to a larger community where the civil society, citizens, patients, representatives are involved from the beginning. So thank you once more for giving me the opportunity to be here, represent what rare diseases are. I think it is important to contextualize rare diseases because it's based on these conditions and how the daily life is for the people living with the rare disease and their family that we based our hope in these new technologies. Rare diseases, as you know, are individually uncommon, but collectively they affect 300 million of people worldwide. All these families, many of them, really experience stigma and isolation. As you can read, for the over 6,000 rare diseases, there are only the 6% of these diseases have an approved treatment. So people are cured. And we see that the percentage that people that will be affected in their life will be of 4%. But what is important, I'm sorry, not very well done, 75% of rare diseases affect children, and the 72% are of genetic origin. Really, they are not number. They are lives. They are people. They are children. They are family. The 30% of children who have a rare disease will pass away before reaching the age of 5. And we all experience what is called the diagnostic pathway. There is an average of 5 years of waiting time to reach a diagnosis. But for some people, this diagnosis won't ever arrive. And they, however, we know that there are people that have been waiting over 30 years to reach a diagnosis. And you can imagine what is the impact of what they went through while waiting and visiting specialists throughout the country or in Europe, even in other countries across the oceans. But rare diseases are not just in health issues. It's not just impacting on the person, but on the whole family, on what we call the caregivers. The 2/3 of the family carers spend more than 2 hours, and this goes back. Yesterday we mentioned the costs, the ratio of cost of having an early diagnosis and an early treatment where possible. And this is another value or another figures that we should add to that calculation, because it's not just the person who is affected and who is diagnosed late in life who won't be able to actively contribute to the life, to the social, economic life of the community at his, her best. but it's also one of the members, at least one of the members of the families, because they have to stop or reduce their professional activity. And obviously this takes and has an impact on the mental well-being. People and their family members, people affected with a rare disease and family members, are feeling depressed much more, 3 times higher than the normal population. And these are results from the Rare Barometer survey, which is a tool conducted by Eurordis throughout European countries. As I said, Eurordis, why it is important that such organization driven by patients that really reinforce the voice of and the rights of patients is important, because we are the safe keeper of this vulnerable population. There are many people who don't have the strength, the time, the strength, the possibility to make their voice heard. And this is why it's so important that patient organizations such as Eurordis are working tirelessly for the recognition of their rights. I said that we are safekeepers. Yes, we are, because while we are advocating for the recognition of the rights of people with the rare disease and their families, so to an equal access to treatment, to a better care, to social recognition, to social inclusion, we are obviously looking at with a huge hope to what is coming from the science. And all the scientific advancements are a beacon of hope for us. So we are welcoming and we are discussing about AI and these new technologies. But obviously we are concerned, and we know, we are aware that they have to be handled with care. So we are starting an internal debate when possible. It's not as multi-voice as it is today, and this will be something that we would love to. We are not there yet, but we will be very soon, I hope. But obviously we know, and these are our key points emerged by our internal debate. Obviously, we want, and we are very much in the hope that these technologies, they are coming, and such as advanced therapies that really can just alter the pathway of the disease. So they will change, they are changing. The few advanced therapies that we already have for rare diseases are really changing the course of the life of the people, of the children, so their own life, and of the families. But at the same time, we are aware of this technology, so the already mentioned CRISPR and CAR-T for rare cancers, for example. We know that we need still to increase our knowledge, and we have to be involved, patients and real-world evidences, and the outcomes of patients should be involved in a collection of important data, but also the perspective of patients should be involved, because obviously we are welcoming all these new technologies, but we don't want them, obviously we don't want to cause harm. We want the choice, the individual choice is respected, but to have a choice that a personal choice, that means that the person has been informed about what's going on, what these technologies are. And it's not easy. So this is why there is, again, a request to find opportunities to open a debate and to engage all the citizens, and not only the rare disease community representative, in this debate. And we are very much aware and concerned about the ethical use of resources. So, because we know that the sustainability of the health system are a key issue at the moment, so we have to know how to use it, and this use should be based on ethical choices. And very important, as I said yesterday, that these technologies should address our unmet needs. And that means that they should be purpose-driven, to really target what we need important for us. Obviously, these technologies are not just for cure, because genomics and bioengineering breakthrough are also tools to detect genetic conditions. And as I mentioned previously, how important it is to have an early diagnosis to prevent a different and more complex path of the disease lifelong. We have what emerged up to now is a general consent on somatic gene editing, but not yet, not ready, but I think this is a common perspective on, we have ethical concern on the germline, which really will generate, we don't know what consequences on future lives. And then still talking about equality. It's our main focus. We were talking about the few treatments available or the quality of care. What we want is that all the people in need are equal access to this treatment. So whenever these new technologies will become available, it's an ethical issue that they will become accessible to them. And I conclude saying that, again, we have a need, a huge need to open this debate, because science is running faster than regulation. The citizens are, as just showed, are not sometimes well informed, or we don't know the sources. So we need this kind of debate where the multi-perspectives are respected, and we all listen to each other in order to create our informed decision on what it could be the use of these new technologies. So the rare disease community, obviously, it will be meaningful, and it's a sound impact on this, but also the general population. So it's really an invitation, and it's a call to repeat this kind of debate and find all the solutions, the opportunities possible, and we will be very happy to host you, to collaborate, to create together opportunities where we can discuss together about these technologies that really represent hope for us. Thank you.
Thank you, Simona. One of the things that I'm very proud of, I'm the president currently of an international society, which is the International Society for Pediatric Innovation. And one of the sister societies, scientific societies, is made by children innovators. Many of them are children with rare diseases, and they actively work for innovations, which is exactly the direction you just told us for now. It's real fun, and I think it's very powerful, and this is a very good example. Questions from the audience? 1 and 2. If we will have time, we will leave some room for questions at the end of the session as well. Please.
Hi, thank you so much for sharing this information. It was very useful. I was just wondering, from an organizational perspective, where in the healthcare system or academia does Eurordis fit in, also in this landscape of European countries having different European healthcare systems? Slightly.
Thank you. Thank you for this question. So, Eurordis as a patient-driven organization, but because we try to do what we are stating and calling for, so collaboration, networking. Networking is the key word for the rare diseases community. So, for example, ERNs, which are the European reference networks, the claim is let the expertise travel, not the patients, okay? Eurordis was so strong to advocate for the importance of this, the creation of this European reference network at the European Commission, that we really made our voice heard so loudly that now we have 24 ERNs, where the collaboration between clinicians and EPEGs, which are the patient's representatives, is very strong. It's organized in different way, depends on the experience of the reference network, but it's very strong. We also have an open academy. It started 15 years ago in Barcelona, the summer school, but now it's an open academy, so winter and summer, where clinicians and academy are speakers, but are also students, and there is a mutual exchange and a point of contact, and also with researchers, because this is one thing that we really deem important, mainly for researchers, not only for clinicians. They know us very well, but for you, just the idea that you see behind what you see under your microscope or your studies, there are people, person with their needs. So this is how we try to create point of contact and opportunities to be and work together.
Last question. Was here? Was one or two? No? There was one, yes?
Yeah, thanks very much. I was wondering, how about representation in these patient groups? We talked about it a little bit yesterday. Like how diverse are they really? Do you see that certain kinds of people tend to participate more than other groups?
Do you mean in Europe or representative of? Okay, so here Eurordis, because I already had this kind of question. Eurordis, as it is for Uniamo in Italy, which is the national alliance within Eurordis that represent the European level, we work together on transversal and common issues. So if Eurordis advocate at European level, at European Commission, the need for the building of European reference network, as well as European action plan for rare diseases, at national level, we will have the national alliance working in collaboration, in coordination with the European level in order to set and to implement what it has been decided, or we are trying to build, or we are advocating for at European level, in the same way, coherent way, at national level. This is a way to lay the ground for the specific patient organization on specific disease. So they are member of the national alliance and then represent at the European level. This represents the umbrella organization, European level, at national level, and then we are supporting with our advocacy on common issues, the action and the representativeness of even the single and smaller patient organizations. Did I answer your question? Yep.
Good. Okay. Thank you so much, Simona, again. Next speaker is Luigi Ripamonti. Luigi is a renowned journalist for Corriere della Sera, which is a very big newspaper. Luigi, please.
Thank you so much. I'm here just to say that Sai already told you why you should beware of me, of course. Well, the point is, from my point of view, I beg your pardon for my English in advance. Okay. The first question I have to ask to myself is information right? Yes, it is right, and it's an issue fundamental for democracy and for freedom. But information is also a commodity. And it is a commodity because it comes at cost of expenses for ink, paper, transport, energy, and so on. But it comes at a cost of people who produce information. So it's commodity, but it is not commodity for free, as we perceive it through thanks to internet from many years to now. This perception has a profound impact on the quality of information. You can imagine we are in the field now where there are no rules. The competitors are everywhere in every way, and everyone try to get attention to readers of listeners, watchers, trying to tailor the information for the desire of these potential readers. And what does it mean in media media reporting in medicine or in science in general? They want to know solutions, easy solutions, immediate solution, free solution, fantastic solution. You can imagine that the production of this kind of news, as Sai told us, can impact not only on individual life, in a life of individual, but on the perception of the veridicity of science in generally. There is a question, as Carlo Casalone wrote in a recent paper, of faith in the science. Well, if we don't trust in what we read, we don't trust in science, and we don't trust in the progress of science, and so on. So it's a big problem for us. As in this, what can we do? One of the function really crucial in this context is the role of gatekeepers. Well, I am a gatekeeper. The role of gatekeeping in the media is the role of the one who said this become a news, this not become a news. Why? Because we have to follow some criteria. What kind of criteria? The criteria are based on experience, and the criteria is based also on tools like bibliometrics rules. I mean, impact factor, H index, good science, good paper. Okay, they are not perfect, but there's something. If I read the paper on New England Journal of Medicine, I suppose that someone read the paper before me, and there are people that know the issue and the argument better than me. So okay, I trust they can do mistakes. All of us remember the Wakefield case of the Lancet. Okay, it happens, but it's not a rule, fortunately. Okay, this is what we can do, but can we do it really? We have very, very, very big pressure. So it's not always simple, because we are in a context where the competition is very, very high. Sometimes it's very, very difficult to make a fact-checking really good, because always it's going to the speed of light. So there is no time. So it's quite difficult for us. And coming back to the role of gatekeeping, well, when I come from I am a physician, and I come from research. I have a PhD in pharmacology. Coming to this to this work as a scientific journalist from the world of research, I was told, well, remember, our duty is to inform, not to form, not to educate, because the education is up to institution, school, state, whatever you want. Well, I try to follow this suggestion, but and during the years, I can say I don't agree, because when we inform, we do form in a way, we educate it, we influence behavior, individual behavior, and when you speak about medicine, about health, it's very dangerous if you do a mistake. So it's a problem, and it's a problem also because now the things are more difficult for another actor. The actor are SEO, search engine optimization. Every big media, and not only big media, has a structure, a group of people who continually say, well, on the in Google Trends as this, everyone, all the people want you speak about this and not about this one. It's very difficult. And there is another problem emerged during a COVID pandemic. I mentioned impact factor, H-index, good scientific paper. But during the pandemic, many journalists discovered the existence of bioarchive or medRxiv. Okay, all of you know what I'm talking about. Well, these scientific repositories are a very good thing because allow the scientific community to share in a very fast way information, and it's good, in particular during an epidemic like that one. Well, there was a need of sharing information, but if you take a paper not yet peer review on a scientific repository and you interpret this like a definitive truth, and you put it on your in the headline of your newspaper, you make it danger, a real danger. All of you remember the case of hydroxychloroquine, or the mixing genes of HIV and SARS-CoV-2. Well, these papers were retired very fast from the repository, but they escaped to the lay press, and that's it. The danger is done. So it's very difficult to do it. And so there is another problem. Many journalists not well prepared, not quite expert, maybe can interpret it not in the right way also the good papers. I mean, if I take a secondary endpoint on a scientific paper, and I think that is the definitive endpoint, I make a danger, of course, and I influence behavior. So all these kind of things makes we danger, as was told. Well, what can we do? Well, I am in my room. I wrote a sentence that is usually attributed to the New York Times. I'm not sure it's so, but I take it as a guide. And this sentence is. Not everything that is true needs to be said, but everything that is said must be true. Thank you for your attention.
So everybody wants journalists like Luigi because he cares about evidence-based medicine, he cares about the clinical trial design and things like this. Unfortunately, this is not the case most of the times. And one of the interesting areas of research is now how information and how perception of the general public depends on social networks, which is an immense area of research. For example, you know that there's now a very strong migration from Twitter to other social networks this was a problem for politics, for information, for medical information, and for education. And this is an interesting phenomenon, and you can also try to get to synthesize some rules, some math rules for understanding the dynamics and the trends, which is very interesting. However, questions for Luigi? 1 and 2.
Thank you very much. I think this is a very important presentation in the what I would call the economy of this conference, in the sense that if there has to be a democratization of the dialogue between science, ethics, and society. Clearly, we have to recognize that information and journalism has a central role in making the mediation between these different realities possible. Now, one thing that I wonder is the place of, you mentioned the reality of truth, you mentioned the function of gatekeeping that the journalist exercises. I wonder whether you could say something about the importance of responsibility, and responsibility toward both responsible science, when in fact responsible science doesn't find the kind of public recognition that it deserves, but then responsibility also toward irresponsible science.
Exactly.
In the sense of becoming a function of critical assessment and potentially disapprobation.
Well, two kind of answers. I start from the second. Well, the better I understood in the years, the better kind of critics of versus a responsibility of science is silence. Not to mention him, not to mention them, and that drives them crazy. Yes, because if you respond, if you critics, you let them to have the chance to respond again, and so on. And it's an avalanche thing. About the first, the responsibility of communication comes also by a responsibility of scientists. And I tell you an example. During the COVID pandemic, one of the problems, at least in Italy, where the talk show, where they took an expert versus a not expert, and that's good in a way. But the problem, the worst problem was an expert versus another expert, saying opposite things. That's confusion, and that is something that make the fate of science really in a challenge. I don't know if I respond. If I answer.
Beautiful. Professor Genazzani.
Yes, I was. Thank you, Dr. Ripamonti. You have touched fantastic points for our daily life. But I was really concerned. 20 years ago, in the world, a group of physicians, which were sponsored by National Institute of Health in the United States, they mention and they make a conference regarding the risk of hormone replacement therapy in women, rising the risk of breast cancer, of myocardial infarction, and so on. Then we had a huge conference at the NIH, and we were 1,500 people. Only two of them, I and another colleague, were speaking against. That was absolutely not true, and for a series of reasons. Then this was circulating all over the world. Hormone replacement therapy was completely destroyed. The pharma company disappeared. In the education of doctors at university, hormone menopause therapy was not considered more a useful tool. Then 20 years after, the publisher of the articles, they accept at the end they were obliged to publish another paper telling they were wrong. But we don't have any press event on that point. You know, it's not that the press was not mentioned, but we have any press event on that point. Then in the world, still now, hormone replacement therapy is something harmful for your mother or for your sister and so on, and for your daughter. Then you know what to do. Whilst it's absolutely the opposite. Then it's in one side the responsibility of the institution to bring the good news, but also once that the news appear to be wrong, it can be then done absolutely in an honest way, can be wrong. I think it's another responsibility of the media to ask for a conference telling, after 20 years, this something is changing to something fundamental in women's life and in daily life of humans. Then why this not rise from inside yourself, the need to ask to the health authorities, to ask to come back again on topics which we have created so important misinformation all around the world.
Well, really,
much less attractive. That's one of the claims.
no, but well, you touched a point that it's very, very important, and we did it. We did it, not maybe on this topic, but in another. Well, I'm coming from Milan, where we have 4 days of talk about many arguments, so much time, so much arguments, and in some of them we did something like that. I mean, cholesterol level. Okay, threshold level of cholesterol is good or not. I asked questions like, why did you told us until now that it's so and now it's different? An example, and so on. Well, we do what can we do? For the issue you point out that well, it's a sensitive issue. When I started to do this work, I was astonished by the level of information about that hormone therapy replacement, and how did it change in the years. And in my on my side, I wrote something about that. Well, why that happened all these years? So that's what I can answer.
Very last question.
Oh, there is a question. No, you told me you didn't
no.
do any question.
I
yet.
told him that I was
time,
going to prepare a very difficult question. No, it's stupid to make you a difficult question because for you it's going to be easy. No, it's only briefly comment that I think as we said yesterday, we have a great challenge because the anti-science movement, they go hand by hand with the populism that we are living, and this is a real risk for our representative democracy that for me is the only way to manage human rights. So, and there are 3 actors there. One is media, the other are experts, which is something different, and citizens. I think that to limit media is not possible and it's not good. It's going to be worse. But to limit experts, I think it could be a good idea because as a journalist, I have the duty of veracity, but veracity is interpretable. But as an expert, I have a strict duty of veracity. Even when you are working in a expert media, for example, in the area of economy, of science, the duty of veracity is different when you are working in a general media. And I think that we can find this week, last week, a good example of this, that we can limit some expression when they are coming, or some opinion when they are coming from experts, because the European Court of Human Rights has dictated a ruling about one case in Austria, an expert giving the opinion about the vaccines. And I think it's important because the European Court says that we can limit the opinion of an expert. And we need to think that we need to preserve our democracy and to allow everything when someone should be prepared to say something, the truth, something with veracity. And I think that we need to work about this. And also the end literacy. Come back to new literacy. We need a new alphabetization because this is terrible because the network. We are not talking about traditional media where you have this council, the board of directors who are thinking about the news. You have people who are preparing and sending a news in 1 minute without thinking about the consequences. And this is a big problem for our democracy and for our bioethics, I think.
Well, I totally agree with you, but the problem to regulate the opinions and the freedom of speech about something is a very big problem and very profound issue.
Luigi, can we go back to this at the end of the session? Because actually, so that we finish this round, and so we get back to the general issues once we've heard all these speakers. Tim Hunt from the Alliance for Regenerative Medicine USA, wherever. I will take your slides. Yes.
Thank you, everyone. While we're loading up, so my name's Tim Hunt. I'm the CEO of something called the Alliance for Regenerative Medicine. I'll get into that in the next slide. I am not a scientist. I am not a physician. I'm a reformed lawyer by training. So I'll duck now or when the questions start. I am also the father of a 19-year-old boy with a rare disorder, a rare neurological disorder called Dandy-Walker syndrome. Let's see, I've got the slides. Great, just 30 seconds on the Alliance for Regenerative Medicine. So we're a nonprofit organization headquartered in Washington, D.C. We've got about 400 members, and they're anything from small biotechnology companies to large pharmaceutical companies. We've got a couple of dozen rare patient organizations that are members, and some of the prominent academic medical centers. And the one commonality that we have is everybody's focused on cell and gene therapy, gene editing, gene therapy, and cell therapy. We have a little bit of tissue repair. So I joke that if someone's got a great small molecule or an amazing monoclonal antibody that can change the world, we don't care. We think that's great, but it's not. We're very modality specific, which we like. We think that allows us to really kind of focus quite a bit on these very, very innovative therapies. We do a couple of big meetings every year. We produce a lot of data. I'll show some data in the next 2 slides. We bring together stakeholders, and we spend a lot of time thinking about how do you modernize healthcare systems, particularly payers and regulators, to embrace these very, very disruptive modalities of cell and gene therapy. And we really think of it as, you know, our focus, our sort of true north is really around the patient community. And I think Simona did us all a great service by talking about the high unmet need. I didn't even know some of those statistics. The 75% of people diagnosed with a rare disease are children, and 30% of them die by the time they're 5 years old. I think if there's one thing I will take away from this conference, is probably those 2 statistics. So thank you, Simona. Here's a little bit of data. This is the first time we've shown this data. It's our 3rd quarter data between the US and Europe. How many approved therapies do we see? 7 approved thus far this year. 5 are still pending that could get approved by the end of the year, so up to potentially 12 cell and gene therapies that could get approved, and a few have already kicked into 2025, but that's good news for 2025 and for the patients impacted. And this is a look across the globe on how many organizations are actually working on cell and gene therapy. So you see across the top, the developers, those are biotech companies, small biotechs, large biotechs, pharmaceutical companies, major academic centers across the globe, almost 3,000 different organizations working on cell and gene therapy. There are almost 2,000 clinical trials taking place across the world. That is great news, I would argue, for these patients, right? A lot of organizations putting shoulder to the wheel, trying to advance real therapies for real patients. And then you can see on the bottom, investments for the quarter were $3 billion. Since 2021, some may know this very well, some may not be as familiar, investment in the sector has really thinned out quite a bit. So when the pandemic started to ebb, a lot of money came back out of healthcare stocks, biotechnology in general, and cell and gene therapy in particular. So we have unfortunately seen a lot of our biotech companies have laid people off, shut down research programs and whatnot, which is really unfortunate, especially for the patients for whom those clinical trials serve. So we kicked off an initiative in January of this year, and I'll get to why in the next couple of slides, really around the importance of cell and gene therapy, ethics, and society. We had a number of organizations, including Children's National Hospital, that are very supportive of these efforts. And a lot of it is because we kept getting asked, and frankly, I kept getting asked some very, very important questions by some stakeholders that are important to us, right? We give these stakeholders special names. We call them patient groups and caregivers. We call them policy leaders, payers, health technology assessment bodies, regulators, religious leaders, bioethicists. Okay, there we go. We're back. And then media and investors. And they ask, guess what? Great questions, right? Like, why are cell and gene therapies priced the way they are? At $2 to $3 million for gene therapy, that's going to raise a lot of eyebrows. Why are they priced like that, right? Can these health systems actually afford these therapies, right? Will only the wealthy countries have access to them? This has been discussed today in other presentations, right? These are all tricky, but a really tricky, thorny one is the ultra rare disease business model is challenged by seeing a patient only one time. If you're really successful and you can intervene one time and provide a durable benefit for a patient, but there's a very small number of patients, it's not always a great business to be in because there aren't enough patients to really cover your research and development costs. It's a very, very thorny issue. For example, clinical trial, and they can only get dosed once. Some very real world things come up. What if my patient gets a low dose, or my child gets a low dose and it's not effective, right? What if it's too high of a dose? It's too toxic, right? Can my child be redosed if it's not effective enough? The answer is usually no. So it's a lot of very tricky questions. And then we've talked about a little bit today and yesterday, a world in which there are some abuses to technology, from unregulated stem cell clinics to Dr. He, the Chinese scientist that brought us the 3 genetically modified twins, right? So how do we think about needed guardrails for some of this, the science? And that led us to thinking, what we want to do is grapple with some of these most complex issues over a few years. And I'll say we picked this word specifically, grapple, not solve. These are not the kind of thing anyone's going to put on a timeline and say, let's get this done by the end of 2025, right? These are very thorny, some of the most complex issues, right? How do we think about pricing and value? And the way I describe it, the important intersection between innovation. Why? Because too many of our patients are suffering greatly, right? So you have to drive innovation, but we have to have access, because if products get approved and they're not available, no one benefits. And how do we make sure that we have ultimately a sustainable model, because the systems can't implode, otherwise nobody wins. And I can talk more about that maybe in the Q&A. How do we ensure the industrialization of the field and how that can lead to eventually global access, right? So that's code word for how do we drive down costs over time? How do we drive up yields? How do we think about modalities that are much, much, much easier? For example, in the future, in vivo sickle cell treatments as opposed to the ex vivo type that we have now. How do we think about this ultra orphan business model, the ultra rare business model with cell and gene therapies? And then we really do need, I am a strong believer, we need some needed and appropriate guardrails for what we're calling frontier technologies. Yeah. Oh, that's a build. Just build it out. So we talked a little bit earlier today about germline editing. It is really back. People may have noticed over on the right, Dr. He has been back in the news with some entrepreneurs out in California saying they think germline editing is a great idea. They would love to bankroll them with Bitcoin and all this other wild stuff that you kind of can't believe. But also people may have seen recently, just in the last few weeks, it became public that back in May, South Africa published some updated research guidelines that appear to allow for heritable human genome editing. They specifically talk about future children, future parents, and things like that, which I think has sent a chill through a lot of bioethicists that have been paying attention to this issue. And then here's a very handsome group over on the right. We had a very nice meeting back in April with a number of people from my community. We had our annual European meeting. We moved it from Barcelona to Rome, and we got together with the Pontifical Academy of Life, had a wonderful meeting for about 3 hours, and we talked about a lot of these specific topics, which I think went very well. And then I'll just close. I think it's always helpful to close with the patient. So this is the gentleman on the right is a man named Jimmy Olehair. I think he's 37, 38 years old. 4 years ago, he had just about given up on his life. He had a very, very rough case of sickle cell disease. He was going through pain crises. He was having trouble holding down a job. He was married. He had one child. He finally got so desperate, he said, I'm going to try getting into one of these clinical trials for sickle cell disease. Got into the trial. It was successful. He felt amazing. Within, I think, a year and a half, he and his family his wife felt so good about where he was. They decided to expand their family. They had twin girls. So now they have 3 children. And then in September of this year, he felt so good, he took up a challenge to raise money for sickle cell disease patients in Africa. And he decided to try to climb Mount Kilimanjaro, 19,000 feet. Now, I didn't know this, but apparently there are guidelines that strongly suggest that sickle cell patients should never go above 10,000 feet because they lose oxygen levels and it's very, very, very rough. So I talked to him, someone we've gotten to know well, I talked to him in advance. I said, you realize this is dangerous. He said, I know, but I'm going with a really good team. They're going to take care of me. So he climbed Mount Kilimanjaro in September of this year and became the first known sickle cell patient to ever do so. So really a remarkable story, and he feels amazing. So, yep. So thank you. So I didn't touch on any controversial subjects, so I know there'll be no questions.
Well, however, as you know, the most interesting stories are from patients, as usual. So this story was so powerful. And remember how many times we have stories coming from patients that help us in doing what we can't do. Because sometimes because of journalists, because not all people have the competence of persons like Luigi, but also because it's extremely difficult. Stories are very powerful. Questions for our team? Come on, there are so many things. And then we have some time, I believe. Yes, we have 10 more minutes just to get back to the other ideas that we discussed with the other speakers and maybe wrap up some conclusions from this session. Randall, please.
So working with technology modalities that are at the forefront, a bunch of these small startups need quite a bit of capital and time in order to bring them forward to help people, and yet investors are backing up. Yeah. So is this a temporary lull? What are the forces driving this, and what's the forecast?
Great question. I would say while everybody's crystal ball has been either wrong or fuzzy for the last couple of years, there is hope that this sort of 3-year headwinds are getting better. And I'm going to talk about that more in a few weeks when we go out to JP Morgan, the big investor conference out in San Francisco. I think there's, here's a couple of, what are some of the reasons to believe, right? Why do we think that could be true? So we see 13 out of the 15 largest pharmaceutical companies by market cap are in cell and gene therapy, right? There's some very hot areas within the field, like cell therapies for solid tumors and autoimmune disorders. I mean, that's about as hot as you get. The cell therapy news, I think, is pretty good. Some of the headwinds have been some of the commercialization challenges, right? Small number of patients or larger markets that I think a lot of us thought would be slow commercially in the US and Europe, but some investors missed that memo going back a few years. Think sickle cell disease and hemophilia. I think they're real markets. There's real opportunity there. Those are very promising, but they are likely going to be reserved for, in the case of sickle cell disease in the US, say 10,000 or 20,000 patients. There's 100,000 patients, right? You're going to get a slice of those. In Europe, it might be, I think there's 50 to 60,000 patients, maybe there's 10,000 or a little bit less for sickle cell disease. So I think those are real markets, but they're going to take a little time to unlock. There are other very good success stories. Zolgensma is available in 50 markets around the world. So a gene therapy for SMA, very effective. It's in Brazil, it's in Ecuador, it's in Egypt, very non-Western, kind of non-typical markets. And I think you'll see some other very successful launches. And some of them are taking place now.
And not to say that this challenge is different according to the countries, to geographic location, because Europe is completely different, for example. Other questions?
Thank you for the talk. So I was interested in the numbers. So when you looked at, you mentioned that investment has been thinning out in the healthcare industry, but yet there's quite a big difference between Europe and North America and Asia Pacific, right? So I just wondered what are some of the main reasons for this, or some of the more less obvious ones?
Yeah, another good question, and some in the room will know this far better than me. I think investment in biotechnology in general has lagged behind a bit in Europe as opposed to North America, for example. When you get to even more speculative areas like cell and gene therapy, you see that tail kind of pull through even more. In In some markets, I'm very familiar with the Boston market, a little less so with San Francisco, you really see a heavy concentration of venture capital firms embedded right on top of the biotech community. That's definitely the case in Boston. And it happens to be right on top of some of the major academic centers. Think Harvard, MIT, the Broad Institute, right? So they are like almost co-located with a very small footprint. And it makes it much easier for the venture capitalists to get to know the senior people in academia and to be knocking on the door and saying, listen, when you have a next version of a gene editing technology, I want to be your first phone call. So it's very, very close community. And I think there's some of that, certainly across Europe, but the concentrations are very large in the US. And I think that helps unlock more academic technology. I mean, certainly academia, in many ways, gene therapy is the great pioneering centers are in Europe. So I don't think that's a scientific reason why some of these things have lagged behind. I think it has a little bit to do with capital flows. Others may have a better view, but that's my quick read on it.
Other questions? If not, I would propose that, let's check if there's any questions from online attendees. No. So we have time for a last round of comments, just one comment for every speaker in the panel, so that I know that Say Ready probably will say that next time we'll call Luigi for an interview, then possibly Tim will speak with Simona and so on, but this is exactly for this reason that we make this panel. Say, do you want to say a very last comment in the light of the discussion we had?
No, I think not, but probably look
I think so. Well, yeah. We have 5 minutes, so if you wish just to wrap up with a general comment about this session, which is about how we communicate.
Yeah, okay, maybe communicate. I guess one thing that I think is pretty clear is that the media landscape changes. I don't know what the future holds for how this will be communicated in the future on many topics, but in science, I think we are also going to have to figure out how to communicate better. And I'm probably not convinced that the simple ways in which we communicate are very receptive for social media. Social media is mainstream media now. Like most people in the United States at least get the majority of their news from social media. This probably explains a little bit of what happens in politics and voting preferences, but that's not going to change, I don't think. And maybe that'll eventually be Europe as well. So we as scientists, putting out a link to a paper on Twitter isn't exactly going to inspire anybody. So I'm not really sure we have a good plan for that yet.
Thank you. Simona, so one last thought. It's about selling stories. You should be the master of that.
Yeah, I'm selling stories. I've got hundreds of stories, and really you have to believe me that among our communities, I've met wonderful people. We are so strong. So my message is, while we are looking for these new technologies to come, and because they are really potentially life-changing, we are doing our best to respect the human dignity and the human rights. And this is something that we can do it all together, because progressing science doesn't mean that we don't take into account the daily life and the value of the lives of every single person living with a disease. I mean, for me, it's a rare disease and their families. And so we have to progress, and we want to be the driving force of all the scientific advancement. This is why we ask to be engaged and involved in this. And I think there is an African saying, actually, that explains very well what my key message could be. If you want to go fast, go alone. But if you want to go far, let's go together. And this is how this broad community should work, allowing listening to each other, mutual perspective, with respect. This is very rare so nowadays. So thank you again.
Thank you. Last comment from Luigi, because actually you recommend some special things to our, as scientists, we are not doing a very great job with you.
You don't need any suggestion by me. I suggest to myself and to all of you, try to cooperate like today in order to improve the human dignity always.
And I think that communication will be much improved if sessions and conferences like this happen more frequently. Tim, some views from your point of view, which is a little bit more business, I would say, how to sell, but in a sense, how to expand your scope and let special therapies reach out all that are in need, actually.
Yeah, I think a lot of people rightly look at, do you see a $2 to $3 million gene therapy? Like I said, it raises a lot of eyebrows, and people quickly go to, is this even remotely affordable? Won't these healthcare systems implode? And I think if you strip out cell therapy for a second, because that's another case, I think Father Marcus did a nice job talking about the pricing there related to existing therapies. But if you look across the globe, in 2030, take all the gene therapies that get approved that can be seen on a reasonable timeline, add them all up, it's about $18.5 billion in 2030. Now that's not nothing, but it's less than 2 drugs approved in the US, 2 global single brands right now, think GPL1s that are coming and getting bigger. It's also about 1% of prescription drug spending in 2030. And that holds true. That data holds true in the US and Europe as well. So it's not nothing. But the healthcare system should be able to take the next 5 years to prepare for 1% of prescription drug spending that will keep a lot of these children alive, I would argue. So happy to talk more about pricing because it is not without controversy, I know. So at the break or over coffee.
That's great. So thank you so much. This panel is over. So we'll leave the floor to the next panel. Thank you again for the discussion. And thanks to the great speakers.